Okay, what is PCR? PCR or polymerase chain reaction is a technique to make many copies of a specific DNA region in vitro in a test tube rather than an organism. PCR refers on a thermostable DNA polymerase, Taq polymerase, and require DNA primer designed specifically for the DNA region of interest. In PCR also, the reaction is repeatedly cycled through a series of temperature change, which allow many copies of the target region to be produced. PCR has many research and practical applications. It is routinely used in DNA cloning, medical diagnostic, and forensic analysis of DNA.
There are three steps of polymerase chain reaction, and it is done in a process called thermocycling. The first one is the denaturing. We will heat the DNA sample so that it will be divided into two single strains. We will heat it at a temperature of 94 till 95 Celsius. The second one will be annealing. In this process, we will lower the temperature so that the DNA primer can be attached to the template DNA. The third one is extending. After the primer has been attached to the DNA, a new strain of DNA will be produced with the help of an enzyme called the Taq polymerase enzyme. This process will need a few hours to be done, or it can be quicker if you use a machine.
Before we get into the application of RCP, we must first know about jet electrophoresis, a technique in which fragments of DNA are put through a gel matrix by an electric current, separating it concise. This is a PCR reaction producing 400 base pair BP fragments. The DNA band, or as you can see there is a line, they contain many copies of the target DNA region, and this is why PCR is important so that we can see it. Now, we're gonna take a look at an example in jet electrophoresis. You receive a DNA sample from a hair left at the crime scene with DNA sample from 3 possible suspects. You'll test whether any of the 3 suspects matches the hair DNA for this marker, and this is the result. You'll see that the DNA from the crime scene has one band at 200 pp and the third suspect has it too. It's not the first or the second suspect as they have a band at 300 pp, while the DNA from the crime scene does not. So, the third suspect matches the DNA from the crime scene. PCR is used in many research labs. For example, in medical applications, PCR was first used to analyze the presence of genetic disease mutations and tissue typing before organ transplantation. In forensic, PCR can be used to rule out suspects in a crime investigation using genetic fingerprinting. And lastly, in infectious disease detection, PCR used to detect HIV, one of the most difficult viruses to detect, and early detection of viral DNA and the subtypes.